Okay, the next two lectures are going to be on the biomolecules. This lecture will be on proteins, and the lecture after that will be on carbohydrates, lipids, and nucleic acids. So here's a list of all of the functions of proteins, and I don't want you to memorize this list, but what I want you to understand is that proteins are very diverse molecules and that they're very important in the functionality of your body. So I have just sort of a list here. So we have enzymes. They act as catalysts to make chemical reactions, lower the activation energy needed to make chemical reactions happen. We find them as defensive proteins in the form of antibodies. They act as hormones. They act as receptors on the surface of molecules, on the surface of cells. And they act as structural proteins, your hair, the fibers in your muscle cells. They act as transport proteins, and they carry things such as your hemoglobin molecule that carries oxygen, and they also act to regulate what genes, determine what genes are going to be turned on and off. Again, I don't want you to just memorize that, but I want you to see that they are very diverse molecules. So if we start with the monomer of a protein, it is an amino acid. And what we find with proteins is that they are made up of polypeptides, which are long chains of amino acids. They're not branched, they're just going to be straight chains. And the all of them have a central carbon molecule. And if I get my little pen going here. So they all have a central carbon molecule. And of course, carbon likes to bond to four things. So on one side, we're going to find the amine group, and that's where they get their name, an amino group. And on the other side, they have the carboxyl group. And remember, that's an acidic group, so that's where we get the acid part of the name. So an amino acid because of these two groups. So we have the amine group, we have the carboxyl group, and then on the third bond will be to hydrogen. Now, this last one is what makes the different amino acids different. So there are 20 different R groups because there are 20 different amino acids. So we can have something as simple as an H to be that R group. Or it could be a ring of some sort, a long carbon chain. It could be a variety of different things. We're going to see those. But what makes the amino acid different from another amino acid is this R group. That's what's important. Okay. I'm going to need to use these. Okay. And when we look at a protein is really the functional unit. So a polypeptide chain is a sequence of amino acids. When we call it a protein, to be like really precise, we really are then talking about the functional polypeptide or polypeptides. So in this case, we have two proteins that are actually consist of multiple polypeptide chains. So the collagen molecule has three that are kind of braided, which is, it's a structural molecule. And so you can kind of see it's like a rope in a way. Hemoglobin is actually made up of 4 different polypeptide chains. So we have these different subunits. Here's one, here's another one, here's another one. If too bad I don't have a better arrow to show you. I guess I could just use my arrow here. And then in these little pockets, they have these little heme groups of iron that are going to then bind the oxygen for transport. Okay, so I'm going to go through the different amino acids. You do not need to memorize the different amino acids, but what I would expect you to be able to do is to look at an amino acid, figure out where the R group is, and then based on the functional groups that you find in that R group, you should be able to figure out what type of properties that amino acid would have. So amino acids have long names. They have 3-letter names, and now more recently with computers, they have single-letter names. So this is arginine, and all 3 of these actually have acidic groups, which make them have basic groups, which make them positively charged. So they accept protons and become positively charged. And so we find these amine groups in these amino acids. And then over here we have the negatively charged. And so here we find those carboxyl groups where they have donated a proton and then became negative. Okay, and all right, so these types of amino acids then would attract each other, right? So a negatively charged R group would be attracted to a positively charged R group, and we're going to see how those interactions play out. There are also amino acids that are polar. So they're not charged, but they act like charged particles. And again, we're going to find oxygens in these molecules. So we have the OH group here, OH group here. We have the double bond to oxygen, and so on. So these makes these side chains polar, and they will interact with water. Here are some cases that kind of don't fit into our little scheme, and like glycine just being just a hydrogen. And then finally we have our nonpolars. And so our nonpolars, we're not going to find oxygen, we're going to find chains of carbon and hydrogens. And so these are hydrophobic amino acids because they do not interact with water because of their nonpolar nature. Okay, and then one important group, and let me go back one more second to this, in cysteine, where is that one? In the last one here? Okay, cysteine actually has a unique property because of this sulfur group right here. Sticking out at the end there, it can actually form covalent bonds with other cysteines. And so what we find is these, what we call disulfide bridges. And we're going to talk about a lot of interactions between these different R groups, but this is the only one that is a covalent bond, which is going to be a stronger bond than some of the others, more stable bond than some of the others. Okay. And the bond can form between these, just like we saw before in dehydration synthesis. And the bond that forms, you're going to learn 4 different names for the different organic compounds. Unfortunately, they all have different names. So the bond that forms between 2 amino acids is called a peptide linkage or a peptide bond. So here we have 1 amino acid, and so remember, it's always going to have an OH, it's lost its H here, so it has the negative charge, and then a hydrogen on the other side. And so these 2 are going to be removed, and where will be lost is water. That's why they're called dehydration synthesis, or sometimes condensation reactions. And now we have formed a bond between these 2 amino acids, so that's a peptide linkage. It's not any different than the bonds that we saw in the last lecture. Okay, so if we put together a whole chain of these amino acids, we have what we refer to as the primary structure of the protein. And it is this sequence of amino acids that's going to determine how that protein is going to be shaped. And with proteins, shape is very important. So how we combine these different 20 amino acids, you can have lots and lots and lots of different combinations. And that's why we see such diversity in this molecule and its functionality. Okay, secondary structures, once we have the chain of amino acids, there's going to be interaction between the backbone, and let me back up again, the backbone of the molecule. So when I refer to the backbone, what I'm talking about is the chain as it's linked together here. So here's the backbone of the molecule. Like that. Okay, so there are you have polar molecules involved in this, and so you can get hydrogen bonding internally within this molecule, and that gives us our secondary structure. So we can end up with an alpha helice, which is just where it's kind of spiral shaped, and this is due to these hydrogen bonds, these attractions, or we can get what we refer to as beta sheets, and that's a folding of that, and I'll show you a model of that in class, which will help you understand it. The next level of structure that we see is called tertiary structure. This is determined by the R groups, not by the backbone. So depending on the properties of that R group, we may get things like disulfide bridges. And like I said, those are actually covalent bonds. We can get hydrogen bonds between the R groups. We can get an aggregation of those hydrophobic chains. They kind of weakly interact with each other, but they kind of don't interact with water, so they will kind of cluster together. We can also see van der Waals forces, which are those temporary dipoles within those hydrophobics. And then we can also see ionic bonds between the charged R groups. Okay, if we lose the secondary and tertiary structure of a protein, we refer to it as denatured. And when it loses its shape like this, when it's denatured, it's no longer functional. So some of the things that can affect this would be high temperature. So obviously we're increasing the energy and we're going to lose some of those attractions, such as hydrogen bonds and so on. pH changes are going to affect those acidic and basic groups, and so that's going to affect those interactions. High concentrations of polar molecules are going to affect the structure of it, because it's going to be interacting with polars and maybe having some kind of a non-interaction with some of the hydrophobic groups that might change how they're oriented. And then finally, the non-polar substances. If you were to put a protein in a non-polar substance, some of them will actually almost turn inside out. So they would maybe, a polar substance in water, which is what normally your proteins are in, a watery substance, the non-polars would cluster together in the middle, and the polars would be on the outside. And if you stick it in a non-polar systemic substance, it can actually completely reverse that. But anyways, these would be non-functional when they lose their structure, but they can renature. And so if the environment changes. And so this could be a way to control the functioning of the protein. You may not need a protein to be working all the time, so it can kind of be turned off in a way and then turned back on again as it's needed. The last level of structure is the quaternary structure. Not all proteins have quaternary structure. Quaternary structure refers to interaction of different polypeptide chains. So if the protein is made up of multiple polypeptide chains, we're going to see quaternary structure. And so you saw the collagen and the hemoglobin molecule, and those would be examples of the quaternary structure. And the interactions are the same as we see in the tertiary structure. They're between these R groups. And so we see those hydrophobic interactions, ionic bonds, hydrogen bonds, van der Waals forces that are holding, affecting that quaternary structure. And here's just a picture of quaternary structure. So because we have 4 subgroups, this molecule has quaternary structure. All right, in the last slide, proteins are the shape of the protein is very, very important. And it is important because it's going to interact. They often interact with another molecule. And this is generally a temporary interaction. So they bond non-covalently with specific molecules. So the shape of that protein is important. So if it doesn't have the right shape, it's not going to fit with the other molecule and it's not going to function properly. And these R groups are important in these interactions with other molecules. So that's the end of proteins. The next lecture will cover carbohydrates, lipids, and nucleic acids. Proteins just take a little more time because they are so important and they are so diverse. So I hope that helped.